Okay. So Endao Han is an associate research scholar in the Center for the Physics of Biological Function, CPBF in short, at Princeton University. He obtained his bachelor degree in physics from Shandong University in China and a master's degree from the University of Manchester in UK. Then he went to the University of Chicago in US for his PhD, and he graduated in 2018. So after obtaining his PhD, he sensed opportunities in leveraging his expertise to understand active and biological systems. So he moved to Princeton as an independent postdoctoral fellow in their newly founded CPBF Center. And in the near future, he plans to dive deeper in biophysics by learning more from his colleague working on microbiology. So with that, thank you for being here, Endao. Please, the floor is yours.
Thank you. Thank you, Professor, for the very nice introduction. And thank you very much for inviting me to share my latest research with you. This is really a quite exciting opportunity. So to start with, I want to briefly introduce my trajectory. So my PhD work was on soft matter physics and with a focus on the rheology of particulate suspensions. So particulate suspensions are mixtures of solid particles and the liquid. And this is, as you can see in the video, this is something you can easily try at home by mixing cornstarch and water with a 1-to-1 ratio. The result is this very viscous white fluid, and when perturbed gently, it flows like a liquid. But when punched really hard, it solidifies very quickly and produces and generates a lot of resistance. So my PhD thesis focused on this fluid-solid transition. Using experiments and modeling, we try to understand why this phenomenon happened. And during my postdoc, my interest shifted from soft matter physics to biophysics and especially active matter. At Princeton, I worked on 2 projects at the same time. One of them is in collaboration with Lai Zhu, who's now an assistant professor at NUS. We figure out a way to make artificial swimmers that are driven by uniform and constant external electric field. And also these swimmers, the phenomena is called Quincke rotation. And using this phenomena, we were able to generate swimmers with multiple forms of motion. And the other one is on the collective motion and mechanics of bacteria. And that's the one I'm gonna talk to you about today. Okay, so the bacteria that we're interested in is called Myxococcus Xanthus, or Myxo. These bacteria, they live in the soil everywhere in the world. They are very safe to human. So it's a good, very good model system for physicists to play with. So each individual cell has a rod-like shape. They're about 7 microns long and half a micron wide. They have gliding motors on their body, which are like little feet that can propel them on a solid surface. So as you can see in this video, on a solid surface, it can move forward. But another thing these cells do is that they reverse. So they don't move toward one direction all the time. So after several minutes, they reverse their direction of motion and move back and forth and like that. So these movies are actually accelerated a lot. These bacteria are actually move quite slowly with a speed of about 1 micron per minute. So it takes several minutes for them to move across one of their body lengths. The one interesting thing about these bacteria is that they are highly social organisms. They So they are a highly social organism. So they can't survive as an individual in nature, and they always live in groups. And their behaviors as a group vary dramatically under 2 different conditions. One is called nutrient rich, which means that there's a lot of food for them to grow. and the other is called nutrient poor, which means they're starved. So in a nutrient-rich environment, the cells want to expand as large as possible. So they become a thin layer that covers the whole surface, as you can see on the left. And when it's nutrient poor, they change the way they move and automatically aggregate into three-dimensional droplets. These are actually droplets of living cells. And these droplets will later develop into what's called fruiting bodies as a strategy against harsh environment. So these behaviors are actually very important for their survival in nature. But the details on how they develop how they move from a monolayer of cells to a three-dimensional structure is still is still unknown. From the eyes of a physicist, this looks like a de-wetting process where the liquid de-wets from a surface and becomes these water droplets. But the special thing about these droplets is that they are active matter. So in active matter, each component can propel itself using a power source, so it distinguishes them from normal materials, right? So on the left are two pretty typical active matter systems. The one on top is microtubules driven by kinesin motors. And the one on the bottom is a herd of sheep. So they actually represent kind of two limits of the active matter systems. The microtubules it's just a material, right? It doesn't make any decision, and all the interactions are mechanical. But for the sheep, they are intelligent. They align with each other because they have eyes, and they decided to do that. So for the bacteria, it's actually somewhere in the middle, right? So these cells, they are closely packed, so they interact with each other mechanically all the time. But also these cells are highly intelligent. Actually, Myxo is one of the most intelligent bacteria in nature. They have developed multiple channels, multiple signaling systems to sense the environment and also to communicate with each other. So to really understand this system using a quantitative approach, it's very important, because then we know how far we can push the physics without considering the intelligence part of the system. Okay, so in the story I'm going to tell you today, we think that at the beginning of this droplet development is completely controlled by physics, right? And then this is how the droplet is generated. If you focus on the region within the circle, you can see how a droplet grows from a thin layer of cell, right? So to get a three-dimensional droplet, you actually start from going from monolayer to double layer. And then as you grow more and more layers, at some point, the whole thing will get smooth and you get a droplet, right? So what's in a monolayer of cells? So let's zoom in and look at what's in a monolayer of cells. So this is what it looks like. So because each individual cell is elongated, we can actually define a director field for this monolayer. So it looks like a liquid crystal, but it's a special liquid crystal because everything's moving. So it's an active liquid crystal, and normally people call it active nematics. So we can define the director field based on the direction of the cells, and if we assign a color for each direct angle, we can actually find regions within a monolayer where all the colors meet. For example, in the regions within these two circles, and these are called topological defects. And at the center of these topological defects, it's impossible to define a director. So in this system, there are two types of topological defects, and you can assign a charge to each of them based on the winding number, and they are called positive one-half and negative one-half defects. So the positive one-half defects look like a little comet, and the negative one-half defect has this threefold symmetry. In recent years, people have found that these defects are actually functional in many biological systems, especially the positive one-half defect. Here are some examples. The first one are neuron progenitor cells. So people found that these defects will cause the cells to accumulate into bigger clusters, and these will affect the later development of the neural system. People also found similar effect in epithelial cells, where positive one-half defects will generate a high pressure region locally, and these high pressure will trigger cell death. And when the cell die, they protrude out of this monolayer. So this is how epithelial cells use defects to generate out of plane motion. People also found in bacteria, you can see the collision of positive one-half defects that actually causes the cells to go vertical, which is shown in this blue region, and this actually significantly decreases the motility of the cells and causes traffic jamming. So in systems like this, when the cells want to move nicely, they actually slow down their speed to avoid these events. Okay, so these defects are functional, but what they do in our system. Here in the video, we labeled out the regions where there's a monolayer, double layer, or hole, which means there's no cell in that region with different colors. And it turns out that the defects, the existence of these defects will affect how the monolayer develops later. It is more likely for a double layer to develop near a positive one-half defect, and it's more likely for a hole to be created near a negative one-half defect. So the director of these cells within the monolayer actually controls how the system will change in the future. But that is statistics. If you look at individual event, actually you see even richer phenomena. For example, here in this video, what we see is a kind of a classic event where a double layer is generated near an individual positive one-half defect right here, right? So a double layer of cells is generated near a positive one-half defect. But not all the positive one-half defects generate double layers. For example, in this video, we see annihilation event between a pair of defects. And during the whole time, the surface is flat and nothing significant happens. But in this video, in the third video, we see a similar annihilation event of a pair of defects. But right after the annihilation, you can see a double layer generated here. And this double layer cell quickly dissolves, right? So statistically, it's more likely for a double layer to form near a positive one-half defect. But the layer formation is not only controlled by the director field. To really understand this, we also need to look at the cell flow and also the mechanical forces in the system. And the mechanical force is the most important thing that we will take a look at today. In a normal fluid, the flow can be driven by external forces. For example, here, when there's a pressure gradient, it can drive flow in normal liquids. In active nematics, for example, a system like ours, we need to add, it can still be modeled as a fluid, but we need to add two more terms. One is a boundary friction. It's because our system is on a solid surface, and this boundary friction has a form like a viscous drag. So it's proportional to the magnitude of the velocity, and also the direction is pointing to the opposite side. And more importantly, there's this active force term. Later, we'll look at this term in details. But for now, we just need to know that the Q tensor is completely controlled by the director field. And with the director field, we can actually predict the form of this active force. And as you can see now, even without any external force, with no gradient of P, for example, you can still drive a flow in the active system because the existence of this active force. Okay, so the theories can predict the active force based on the director field, which is this Q tensor, and then the active force can generate flow. So normally, when people go and test these theories experimentally, what they do is they measure the director field and they predict the flow using the theory, and then they measure the flow and to compare with the prediction. Because measuring direct field and measuring the flow are much easier than measuring the active force. So what do we do here is we want to test the whole thing by measuring the active force and other forces in the system. So we can test the theories systematically. Okay, and this is like the first time that this kind of measurement has been performed in systems like that. And the technique that we use to measure the forces is called traction force microscopy, or TFM. The idea is very simple. So we prepare a soft elastic substrate with embedded fluorescent particles and put the cells on the surface of the substrate. When the cells apply a force, the substrate will deform, and then these fluorescent particles will move. Okay, so by tracking the motion of these fluorescent particles, sorry, is there a question? Okay. So by tracking the motion of these fluorescent particles, we can measure the displacement of the substrate, and using the displacement, we can reconstruct the force applied by the cells. Okay, so to do that, what we need to know is a Green's function, which is the displacement generated by a point force on the surface of a solid. And to with a Green's function, we can calculate the displacement given any force field by calculating the convolution. And normally people do this in the Fourier space, so the convolution becomes a product. But to know the opposite, to know the force from the displacement, naively we can just put the Green's function on the other side. But actually, this doesn't quite work because the inverse problem is very sensitive to the noise in the system. For example, here we generate some artificial result, and these are some forces applied on a surface within these circular regions. Right? We can calculate the displacement generated by these forces and adding a 1% noise in displacement, which is barely, it's not even visible by naked eye. But when we do the reconstruction, we can actually see, if you use a naive method, the reconstructed force is completely covered by the huge noise in the reconstructed force. So what we need to do is to do regularization. So there are several methods to do that, but the basic idea is to suppress the results that doesn't make any sense and get a reasonable result using these regularization parameters. But the point is, these measurements are very sensitive to the noise. So getting rid of noise, reducing the noise in the system is a key to the success of this project. So TFM is not a new method. People have been using this to measure forces generated by mammalian cells for a long time. But for this to work on bacteria, which are much smaller and also much weaker, they generate much less force. We actually need to gradually improve the capability of these techniques. We kind of push these techniques to the limit so we can measure these forces. Okay. So this is what we did. So we prepare a very soft hydrogel called PAA, and we put fluorescent particles in it. And then we coat the surface with chitosan to enhance the cell substrate interaction. And then we put the cells on top of these hydrogel. One thing to mention is that we put nothing on top of the cells. So on top is just a gel, cell, and air interface. Okay. And when we image the system, we use two different channels. We use bright field images to look at the cells, as you can see on the right, which are these white bars, and also we use laser images to see the fluorescent beads. Okay, so here, as you can see in this region with dilute cells, the cells apply a little force on the substrate, so there's barely any particle deformation. But here, under this monolayer of cells, they actually generate a large amount of force, so which causes a lot of deformation and motion of the particles, as you can see here in this region. Okay, and with these two parts of the information, we can actually get a lot of results. So with the bright field images, we can measure the director field of the cells and also locate the topological defects. At the same time, we can use the bright field images to measure the velocity of the cells and and to know the cell flow. With the laser images, we use a PIV algorithm to track the motion of the particles within the substrate to measure the displacement. And using the displacement, we can reconstruct the traction using the method I just showed you earlier. So one more thing to mention is that even though it's called a traction force, the traction is actually an aerial force density. So they have the unit of Pascal, which is like a stress. Okay. So now with the measurement, we get the director field, the velocity of the cells, and also the traction, the forces that they generate at the same time. Okay. So the first thing we do is we can follow each individual defect, right? So for example, here we follow this positive 1/2 defect, and the orange arrows are the traction within this region. And we can reorient the traction that we measured and calculate the average traction near a positive and negative 1/2 defect. Okay, and it turns out that these tractions do have a pattern near both defect. For example, there's an area with stronger traction pointing forward in front of the positive 1/2 defect, and for the negative 1/2 defect, we have these 3 lobes near in these 3 directions. Okay, so the average traction does have a pattern, but is this the pattern, the active force? So to understand that, firstly, we need to go through some details in theory. So what the theory says is that the active force is controlled by the director field, and the director field has 2 parts. One part is a scalar order parameter that tells you how ordered the directors are locally. In a random in a region where the directors are random, you have a small s, and in regions where the directors are completely ordered, you get a large s, and s is in the range between 0 and 1. So the second part is this matrix that's completely decided by the director angle, the average direct angle everywhere. For example, here, we can see a negative 1/2 defect here, and within near the center of this negative 1/2 defect, the director is not well defined, and S approaches 0, as you can see here. So the blue, so the light blue means 1, and dark blue means 0. So using the scalar order parameter S, we can easily identify all the defects in the system, and then in most of the regions, except for the defects, the cells are highly ordered. So S is very close to 1, and then we can use the local directors to define the Q-tensor. Okay. And then what the theory says is that these director field, this Q-tensor will generate active stress within the system. For example, if you have a region where all these directors are horizontal, and they also want to expand in the longitudinal direction and shrink in the perpendicular direction, it will produce a positive active stress. So an activity parameter is defined to label the sign and magnitude of this active stress. For example, when the system does this, it's called an extensile active nematics with a zeta larger than 0. And when in the opposite direction, when the stress goes like that, the system is contractile with a zeta that's below 0. Okay, so these active stress will generate instability in the system that causes the directors to deform. And when there are deformation in the director field, the divergence, you'll get a non-zero active force, which is the divergence of these active stresses. So normally people assume that the activity parameter is a constant, so you can take it out of this divergence. So now the active force is completely controlled by the director field. In 2D, there are two ways of deformation. One is called bending, and the other is called display. For extensile system, the active forces generated are pointing to the left. So you probably have noticed that by combining these two, you actually get a positive 1/2 defect. Actually, we can label out, on the positive and negative 1/2 defects where the deformation is bending and where deformation is display. And it turned out that for the positive 1/2 defects, these two active forces, they add up. And for the negative 1/2 defect, because of the symmetry, these forces actually cancel each other. So the result is that when you have an extensile system, the positive 1/2 defect will propagate forward. And the negative 1/2 defect will be much less motile, and they normally just move in the nearby region. Okay, for a contractile system, the force goes opposite. But our system is actually an extensile system. In our system, the positive 1/2 defects propagate forward. Okay. So based on the director field, we can predict the active force that should be generated and compare that to the actual traction that we measured. The measurements are on the left, and the predictions based on the director field are on the right. And as you can see, the patterns are not exactly the same, right? So this pattern is more on one side, but this pattern is more symmetric, similar to the negative 1/2 defect. It's more clearly if you only look at this line, the data along this line, right in the middle, as we show on the right? So the measurement is more anisotropic compared to the prediction for both the positive and negative 1/2 defects. And the curves do not overlap at all. So there's something that we're missing. So what we're missing. So to know that, we need to look at what we're actually measuring. So we are measuring the traction, which is the cell-substrate interaction, which does not represent the active force completely. Okay. So firstly, we need to look at the force balance. Because this is a very small system, the Reynolds number is very low, so inertia doesn't play any role in the system. As a result, we have a force balance everywhere. There are two parts in the force balance. The first part are the active forces, and the second part are the passive forces. So as I said earlier, these cells, they move via the gliding motors, and these gliding motors can move on a solid surface. And also they can allow the cells to move on each other, right? So there are two parts in the active force. One is a self-substrate interaction, Fs. The other is a self-cell interaction, Fc. Okay. In the passive forces, there are two resistant force due to the friction between the self-substrate, Fs, and also the self-cell friction, Fc. Lastly, we have a hydraulic pressure term, gradient of P. Okay. So these forces are balanced. But what we're measuring is a self-substrate interaction. So the traction is actually equal and opposite to the sum of the active force, the active force and the resistant force between the cell and substrate. So finally, we got this expression where the force we measured, it actually related to the cell-cell active force, the cell-cell resistant force, and the pressure term. When we do the average, the middle term is actually negligible. So what we measure is not only the active force, but also the pressure term. Okay, so we can decompose the measured traction by separating these two terms. And now, as you can see, after separation of the terms, the active force becomes more symmetric. Actually, it agrees with the prediction of the theory very well, as you can see shown by the black curves. So what's interesting is this one more term that has something to do with the pressure. This is isotropic. With a gradient of P, we can actually do the integral and calculate the value of pressure near the positive and negative 1/2 defects. And it turned out that there's a positive pressure near a positive 1/2 defect, and there's a negative pressure near a negative 1/2 defect. And this is very interesting because a high pressure region is more likely to push the cells to move out of the plane, right? So that's a very good idea. But when you think about that a little more, you'll figure out that this pressure is not large enough, right? Because to move out of a plane, you need to overcome this barrier applied by the surface tension. As I mentioned at the beginning, we didn't put anything on top of the cells, but at the cell-gel air interface, there's always this water meniscus that's confining everything, right? So to go out of this plane, you need to deform the water layer enough to overcome this barrier set by the surface tension and become a double layer. And is 2 Pascal enough to push the cells out of the monolayer? And the answer is no, it's not even close. So we don't know the exact number yet, but using calculation and experimental measurements, we estimated that this number should be somewhere, it should be at least tens of pascals. So 2 Pascal is way too small to make this happen. So it's not a uniform, so it's not the average pressure that make the cells to move out of this plane. So what do they do? What's the cause of this movement? So actually, these are the average traction that I showed you earlier near the positive and negative 1/2 defects. But these are calculated using the vectors of the traction. So now, if we look at the magnitude of these, the average magnitude of these traction, we see a very different numbers, which is almost a number, order of magnitude larger than the average using the vector. Okay, so this means that in these regions, there are strong fluctuation in forces. The forces actually point in random directions with very large magnitude. So when we calculate the average, the vector mean, most of these forces cancel each other. What's left is not zero. It has a trend, but it's much less than the fluctuation itself. Okay. And what's more, we found that these fluctuations in traction is less dependent on the director field. For example, here, the most deformation happens near these defects, right? And in regions where there's no ordered cells, there's no defect, the cells are very well ordered, and based on the theory, there should be no active force. But instead, in our measurement, we see very strong force fluctuation in regions even where the cells are highly ordered. And this is something that's been kind of ignored before by both theorists and experimentalists. So the active force is not only generated based on the director field, due to the curvatures in the director field, but there's also a strong fluctuation that's independent of the director field. So here we plot the distribution of the traction magnitude within these circles near the positive and negative 1/2 defects, and also within regions where the cells are highly ordered. As we can see, in all these regions, there's a wide distribution in the traction magnitude, but also there's an order, right? So the positive 1/2 defect has a wider distribution compared to the order region, which has a wider distribution compared to the negative 1/2 defect. So this is interesting. So even though the average force is not large enough to cause the cells to go out of the monolayer, the positive 1/2 defect still generate the strongest force fluctuation among all the regions. But this difference is not enough to explain why there's so much higher chance for a double layer to occur near a positive 1/2 defect. We also need to understand that we also need to look at the velocity in the system, which is the cell flow. So we track using the bright field images. We track the cell motion using a method called optical flow, and so we can measure the velocity in the system. And similarly, we calculate the average velocity near a positive and negative 1/2 defect. And it turned out that there's a net influx near the positive 1/2 defect, and there's a net outflux near the negative 1/2 defect. So if you look at the illustration underneath, for the positive 1/2 defect, there's actually more chances for the cells to, the cells will have more chances to move out of the plane because there's a strong velocity in the tail of this defect, but they're blocked in front. So when there's a net influx, the local concentration of the cells will increase, so the cells want to go outside. So these double layer generation events are very similar to the nucleation process. So you have this frequency of the attempt, but there's a energy barrier between you and the lower energy state, right? So you are making this attempt with a certain frequency, and when you get a high enough energy, you go over the barrier, right? So here, the energy is basically the stress, and the frequency of attempt is controlled by the velocity field of the cells. We actually need both a high enough stress and the net cell influx to generate a double layer, right? And when we look at the chances, the positive 1/2 defects are always larger than other regions than a negative 1/2 defect on both of these two categories. That explains why it's more likely to generate a double layer near a positive 1/2 defect. And you actually need both a cell influx and a high enough stress to achieve that. Okay. But let's go back to the functions of the cell, right? As I said, when the cells feel starved, they aggregate into these clusters. And what do they do to do that? They actually turn off their reversal, right? So in a nutrient-rich environment, the cells move back and forth. But when they feel starved, they actually move more ballistically by getting rid of reversal. Okay, so how does that have anything to do with the cell influx and the stress? So to test that, we use the mutant that is genetically modified, and so that they don't reverse. And we compare that with a normal cells that we use that do reverse. So on the left are what you're seeing here are the cells that do reverse, and on the right, what you see are the cells that don't reverse. Okay, so the difference is very significant. On the left, you see more flat regions where the cells are within a certain plane. But on the right, one thing is you see much higher concentration of the defects, and also you see regions where the cells are kind of out of focus, which means they're aggregated into three-dimensional structures. Okay, so by turning off reversal, you actually the cells will automatically aggregate, and we measured the distribution of the speed of the cells and also the distribution of the traction magnitude. It turned out that when getting rid of reversal, the cells don't move faster at all. So the distribution of the speed are exactly the same. But as you can see on the right, the traction that they generate is way larger than the cells that do reverse. Okay. And another thing we looked at is the flow near the positive and negative 1/2 defects. So here are the what you saw earlier. These are the flow fields generated by the cells that reverse, and on the bottom, these are the flows generated by cells that don't reverse. So I made the scale bars the same, so you can see the comparison, right? So even though the average speed of the cells don't change at all, the average velocity near the defects increases when there's no reversal. And also the width of this flow that goes in and out of the defect region also expands, so which generates a stronger influx near the defects. So by simply turning off reversal, you actually increase both the cell influx and the stress fluctuation at the same time. And the cells know that somehow. So that's exactly what they do in nature. Okay, so to sum it up, we developed a method to measure the director field, the velocity field, and traction at the same time in a thin layer of Myxococcus Xanthus. By measuring the traction near the defects, we find that the average traction does have a pattern that can be explained by the actin nematic theory. But at the same time, we found something that's been ignored before, which is a very strong traction fluctuation in such systems. That's much stronger than the mean, both near the defects and in order region. Lastly, we want to relate what we measure to the function of the cells, which is to form double layers and move out of the plane, right? And we found that double layer formation needs both strong enough local stress and also influx of cells. And getting rid of reversal promotes both at the same time, thus creates cell aggregates. So I want to end with these pictures on the right. So these are generated by relatives of Myxo in nature. Okay, they actually have really rich and beautiful shapes. So for the Myxo is one that can survive in the labs. Most of these people don't know how to keep them alive, so you can only find them in the nature. But I think the message is when biology meets physics, it can really generate a lot of beautiful structures. And in nature, there's a lot of mysteries out there waiting for us to understand and to discover. Okay, here I want to thank my collaborators. Josh, Katie, and Matthias are experimentalists. Ricard, Chen-Yi, and Ned are theorists I work very close with. I also want to thank the CPBF for funding. And also, I'd like to thank you for the invitation and for listening. I'll leave my summary slide here and I'd like to answer. I'm happy to answer any questions. Thank you very much.
Thank you. Very nice for these talks. Okay, so open for questions. Massimo?
Yeah, perhaps I can start.
Sure.
So thank you very much. I really enjoyed the talk, and also this idea of connecting the behavior of physical system to topological defects. I find it fascinating. One of the questions that I have is the role of replication. So is the density of bacteria constant in your experiment, or they are duplicating? This would be another effect that is leading to, I suppose, 2 to 3D transition.
Yeah, so that's a very good question. So in this condition, it's when we do the experiments, it's a nutrient-rich environment, but the temperature is lower than the temperature needed for them to reproduce. So the number is kind of a constant during the whole process. Some cells will die, but it's roughly a constant. So the cells double at 32 Celsius, but when we do experiments, we actually do it at the room temperature, so they don't duplicate. And also every time we try to put we try to control the cell concentration that we put on. So it's roughly a constant.
Roughly constant. Okay.
Yeah.
Thanks. The other question, it's more fundamental, because you have been trying to validate a continuum approach to describe the behavior of this system. Now, in a continuum approach, the basic idea is that you are cross-grading the system usually using a size scale or length scale that should be much larger than the size of the individual element, right?
Yes.
So in this case, I'm just wondering, when you develop, I don't know, when you measure the traction on some typical scale, how does this scale compare to the size of the bacteria? You have sub-bacterial resolution or not? Yeah.
So currently, our the limitation in the resolution is basically our experiments, right? So we can't use we can't make the interrogation box too small. Currently, our interrogation box, our resolution of the traction is about 1 micron by 1 micron per box, and our cell width is about half a micron, right? So each individual box is wider than a single cell, but also the cell length is about 5 or 7 microns. So it's much shorter than cell length. So this is kind of it may not be ideal, but it seems that for now everything works by assuming the cells are just assuming that the system is a continuum system. One possibility is that the cells are actually very soft, so they even it's a long cell, they bend pretty easily. So it seems that the mean field approach works pretty well, both in predicting the velocity field and also in the traction field. But yeah, currently the limitation is mostly from the experimental side. If we use smaller particles and higher particle concentration, we can probably shrink the box size and make higher resolution traction force. But also, as I said earlier, the things can be ruined very easily by the noise in the measurement. So yeah, there's a balance.
Related to this question, you show that you have a very important rule of fluctuations that are not captured actually by the continuum equation. So yes. Do you think this is actually a warning towards the trend in developing a continuum approach for active matter, in the sense that possibly the fluctuation is so much larger that this continuum approach may generally fail, like essentially you have been showing?
Yes, that's a very interesting question. We actually have some speculations. It doesn't go against the continuum theory, but my supervisor doesn't allow me to show anything that we're not sure yet. But yeah, we do have some speculation. We think that there are two possible reasons why there's such a strong fluctuation. One of them is a cell-substrate interaction. So in many active systems, right? So there's only interaction between component and component. But here, these cells, they not only interact with each other, but also they interact with the substrate, right? And when they interact with the substrate, it generates some extra propulsion that doesn't care about the director field, right? So the cells just want to move forward. And when these cells collide with each other, it can generate locally high pressure regions, right? So that's one major source of the fluctuation. The other one is also kind of ignored in the theories. So that's where when you expand the divergence. So here, normally people assume that activity is a constant, right? So when they expand this, they just take the activity out. But in real systems, we don't think it's the case, right? So especially in our system, there's no guarantee that activity is the same everywhere, and it's very likely that you actually have a distribution in the activity in the system. So when you expand this term, you not only get this, but also you get Q dot the gradient of activity, and that can also that can give you perpendicular forces in every region, even ordered region, right? So for example, if you have an ordered region and you have a gradient in activity in the horizontal direction, that can actually give you a horizontal force. So yeah, so these two are possible reasons that we think can cause these large fluctuations. But we're trying to test. We're trying to use our data to test these, but we haven't really done that yet.
And perhaps the very last question, sorry, I'm asking too many questions, concern the distribution of the traction force that you have, I think is the next slide or in a couple slides, when you compare two strands of the bacteria, the original one and genetically mutated. So the fact that in one case the distribution has this very large tail when you study the distribution, this is due, I don't know if the guess is right, is because you have some kind of alignment, so that bacteria that want to propel in the same direction, they are close to the other. This is why. So in local region, you have all forces that are parallel, and so they adapt. And you measure that force, or there is some other physical mechanism?
Yeah. So we we haven't really figured out the detailed mechanism yet. But I think that the idea, the general idea is when the cells reverse, it actually like a mechanism to release the local high pressure, right? So it doesn't seem that the cells care about local pressure, but it's not like when they see a high pressure that increase the chance that they reverse. The reverse seems to be independent, but nonetheless, the reverse can release some of the stress within generating in the system, because when they just reverse, the high pressure will not be there anymore. But when they don't, and they move ballistically. It's also they move in bigger flows. It's more easily to generate regions with high pressure that doesn't release, right? So this is like the general picture that we've been having, but yeah, the details are still missing. Yeah.
Okay, thank you so much.
Oh, thank you very much.
Thank you, Massimo. Other questions? So I have a question myself. I mean, I'm not specialist at all in this domain, but I see this. I mean, this topological defect has some quasi-particle in the condensed matter language in some sense, right?
Yeah.
Where the topological charge will be charged. So in the situation of with quasi-particle, it exists some interaction between these particles. I mean, you should take, for example, two charges, right? Electric charges that will be attracted, or so that there is some interaction between these. Is it the case here? I mean, since there are, of course, this difference of pressure, so there should be there should have some forces between those two, between those particles. They are annihilated as well. I mean,
yes.
So can you? I mean, did you observe this? I mean, do you have any proof of some kind of interaction? Is it short range or long range interaction? This kind of thing?
Yeah, so that's also a very interesting question, but we're not there yet. So there are definitely some interactions, and those are predicted by the theory. But currently we are only focusing on individual defects. So the data for the data I showed you, those are like defects that are kind of away from other defects. We want to look closer to the look closely to the interactions later. So now I don't know much about it, so sorry. but in this case, we only focus on individual defects that are away from the others. But yeah, there are certainly interactions. So there are, for example, when two defects approach each other, there's actually preferred directions. For example, the positive one-half defect is more likely to come from this side instead of this side. So it's kind of controlled by the director field. It's kind of decided by the flow. Maybe also there's forces involved in it. But yeah, so we actually have a rich amount of data. It's just now we're trying to get more and more instances from that. But we're not there yet. But thank you for the suggestion.
I see. So that there is some kind of anisotropy also in the interaction, probably.
Yeah. There's definitely some. Yeah. So there's the most important. Okay. So the most obvious thing is that yeah, there's definitely some chances for the some probability distribution for the defects to approach each other, and also there are always a preferred direction for the defects to approach each other. For example, if the positive defect goes this way, the tail will be like that instead of like this, right? So and also another thing that similar to what you just mentioned is the interactions between these positive and positive one-half defects. For example, here in this system, a very large force can be generated when two positive one-half defects collide with each other, right? And it's kind of intuitive, but we saw all these events in our data, but we haven't had the time to analyze them yet. But yeah, this could be a very interesting question, indeed.
I mean, also, your label, your defect as one half and minus one half. Why not 1 and minus 1?
Yes.
Does it mean that there is other categories of defect that you didn't observe yet? Or?
Yes. So our system is a nematic system, which means each individual element has a direction, but it's not polar, right? So if you look at the director field, the range of this angle is only between 0 and pi, because theta and theta plus pi are exactly the same, right? And in a nematic system, the positive and negative one-half defects are the only stable ones. you can see +1 and +1 defects, when you have a polar system, which is more like the sheep, right? So in a polar system, each component does not only have a direction, has a director angle, but also they have a preferred direction of motion, right? And also these little arrows will want to align with each other. And in a polar system, you can actually have stable +1 defect. But in active nematic systems like ours, the +1 defects are not stable. It'll kind of split into 2 +1/2 defects, and they will propagate away.
Okay. I see. Very interesting. Okay. Any other questions? Okay, so if not, thank you very much, Endao, for the talks, and sorry for this very early time for you, and all the best for the NAP. Hope to see you in Singapore.
Thank you very much.
Goodbye. Bye bye. Thank you.
Bye. Have a good day.
You too.